There are several challenges associated with standardizing biomarkers for the diagnosis of mesothelioma. Mesothelioma is a rare form of cancer that develops from the mesothelium, the protective lining that surrounds many of the internal organs of the body. It is commonly associated with asbestos exposure. Some of the key challenges in standardizing biomarkers for mesothelioma include variability in biomarker expression. Mesothelioma tumors can exhibit significant heterogeneity between patients in terms of molecular and protein biomarker expression profiles. Different tumor cell subtypes and molecular phenotypes have been described for mesothelioma that are associated with variable biomarker expression. This intrinsic biological variability between tumors makes it challenging to identify biomarkers that are reliably expressed across all mesothelioma cases. Biomarkers that are only expressed in certain subtypes may have limited utility as standardized diagnostic markers. Lack of large prospective validation studies. While several candidate mesothelioma biomarkers have been identified in retrospective case control studies and smaller clinical trials, there is still a lack of large, high-quality prospective validation studies to definitively establish the diagnostic accuracy and clinical utility of potential biomarkers. Prospective studies following at-risk asymptomatic individuals over time are needed to validate that biomarkers can reliably detect early-stage mesothelioma prior to the development of clinical signs and symptoms. The rarity of the disease makes conducting very large prospective validation studies challenging. Complexity of biomarker panels. Most studies suggest that no single biomarker will have sufficient accuracy for mesothelioma diagnosis and that biomarker panels incorporating multiple analytes may be needed. However, determining the optimal combination and testing algorithms for biomarker panels can be difficult. Different biomarkers may also have varying stability, specificity and sensitivity profiles depending on tumor site, stage and other clinical factors, complicating the standardization of panels. Extensive validation work is still needed to define highly standardized and reproducible biomarker panels. Pre-analytical and analytical variability. Ensuring consistent, reproducible results for biomarkers across different testing laboratories and settings poses additional challenges. Biomarkers can be affected by pre-analytical variables like specimen handling, processing and storage conditions which need to be tightly controlled. There is also analytical variability inherent in different biomarker detection platforms, reader technologies and laboratory practices that must be minimized through standardization efforts to ensure reliability and comparability of results in clinical care and research settings. Lack of standardized reporting systems. Currently, there is a lack of consensus on standardized systems for clinically reporting mesothelioma biomarker results. Issues like appropriate cutoff values, classification categories and descriptors for biomarker positivity need to be agreed upon. Without standardized reporting frameworks in place, it is difficult for clinicians, patients and researchers to properly interpret, compare and utilize biomarker data across different diagnostic settings and studies. Heterogeneity across subtypes. The three major histological subtypes of mesothelioma, epithelioid, sarcomatoid and biphasic, exhibit distinct gene expression profiles and biomarker patterns. Current biomarker research has predominantly focused on epithelioid mesothelioma. However, markers validated in epithelioid disease may have reduced accuracy for sarcomatoid and biphasic subtypes. Standardized biomarkers would need to demonstrate sufficient efficacy across all subtypes. Lack of regulatory approval. To date, no serological or molecular biomarker has been approved or recommended for standardized clinical use in the diagnosis of mesothelioma by regulatory bodies like the FDA in the United States. Further demonstration of analytical and clinical validity through large prospective studies will be required to achieve regulatory endorsement and reimbursement, facilitating wider spread clinical adoption of standardized biomarkers. Overcoming variability in biological expression. conducting extensive prospective validation work, standardizing complex biomarker panels and testing methodologies, improving pre-analytical control and developing consistent reporting frameworks are some of the key challenges that must still be addressed for the successful implementation of standardized mesothelioma biomarkers into clinical practice. Continued biomarker research and standardization efforts are needed to advance early detection and diagnosis of this asbestos-associated malignancy.